So now is the big question. How does an adenovirus-based vaccine work? We know how does a messenger RNA-based vaccine works, and we know about the lipid nanoparticle and so on. So let's see how does this work. So here is the virus itself, adenovirus. And what happens is, if I can show this diagram here as well, this diagram, if you see here on this virus, see those little red and green things? So on top of the virus, there is a coating of proteins, and you can call that sort of an envelope, just like SARS-CoV-2 has an envelope as well. So here I have made that in green. So we have a virus particle which is coated in some proteins. This virus particle has these red spikes on it, just like SARS-CoV-2 has spikes. Here we don't call them spikes, we call them fibers. So these fibers connect with our cells and receptors. Just like spike protein on SARS-CoV-2 binds with our cell on ACE2, these spike proteins also bind with some other receptors on our cell. The result of that is that this adenovirus is brought into our cell inside a small vesicle. Vesicle will be a small piece of our cell membrane that becomes a pouch around this virus, and the virus is brought in the bubble made up of our cell membrane. So far so good. We have a virus. This virus, especially in case of AstraZeneca, the vaccine, this virus is made in a lab, and it is made in HEK293 cells. We have discussed this many, many times before. The exact production of this virus has been discussed before. The exact function has been discussed before. The comparison with other vaccines has been discussed before. So I'm not going there. I just want to make sure that we can understand, once the virus enters our cell, what's going to happen next. So here is the virus particle, connects with the receptor on our cells, and then it is brought in a bubble, which we call vesicle, which is made up of our cell membrane. This is just like if you look at my hands here. So imagine if this gray cloth here is the cell membrane, then when the virus is entering this cell membrane, it takes a piece of the cell membrane with it, and it is covered by that. So that is this structure here, that blue thing around it. Now, what we do is, and we have discussed this in the past as well, that when we bring something from outside, that is called phagocytosis or pinocytosis or endocytosis. When we bring something from outside, we then combine that phagocytic vesicle with the acidic vesicle that we have in our cell. You can say small stomachs are present in our cell. These tiny microscopic stomachs, they have lysosomal enzymes in them. Lysosomal enzyme simply means they have chemical substances that would digest and destroy whatever they come in contact with. So we fuse the viral particle vesicle with the lysosome together, which allows the lysosome to spill its guts, its acids, in to this virus particle vesicle. When that happens, because these tiny stomachs they have acids in them, so what would happen is that as the acid appears in this vesicle, the pH drops. pH drop simply means acidity inside this cell the vesicle increases. So far so good. We brought in the cell. So folks were complaining yesterday that I was going fast, so I'm kind of making sure that we go step by step. So the virus brought in a vesicle, then we fused with that vesicle the lysosomal vesicle, which is a small stomach of the cell, and there are millions of these stomachs inside the cell. They have acids in them. Those acids are brought into the vesicle. Now, the adenoviruses are very clever virus. If this was another virus, bringing in the stomach or the acids will actually destroy this thing. But adenoviruses, the common cold viruses, have changed themselves that when they get this acid, it allows them to become active. So imagine these viruses look like an orange, which has a skin on it. And imagine if if you put acid on that orange, that would cause the skin to be removed and the fruit to come out. That is what these viruses have adapted themselves as. So when you try to destroy them, they actually become active with that. So what they do is, if you see here in this part, they remove, using those acids, they remove their coating and they remove the fibers that were attached to them. The remaining thing, this is called uncoating. They remove their coat. If I took my shirt off, that will be uncoating. So they remove their coat. And now what remains is the virus DNA and the proteins around that are called capsid. So very sharp virus. It used the acids to remove the outer layer. Now this inner layer that comes up, that is available, that can escape from this phagolysosome. So that is the clever thing about this virus. So virus can then break out of it. This is called the viral escape. Isn't that interesting? Other viruses don't do it. Some viruses do this. So when it escapes out here, so see this little red thing. Now this red thing has removed its coat, it has given up its fibers, and the capsid and the DNA has come out. Now in this DNA here, in this DNA is the genetic material for the spike protein. This is a virus whose DNA we have changed in our labs, that this virus cannot make more viruses, and all it contains in it are the genetic material for the enzymes to help replicate its or make messenger RNAs, and the messenger RNA for, or the DNA for the spike protein. So keep an eye on this guy now. The thing is that this guy here, it has to actually send its nuclear DNA, its DNA, not nuclear DNA, its DNA, which contains the DNA for the spike protein, in the nucleus of our cell. Because DNA can only be converted into a mRNA inside the nucleus. So that is this virus's challenge. So you may ask me a question over here, and that is, well, we had always heard that the spike protein is RNA. Why do we have a DNA of the spike protein? So in the lab, we manufacture the DNA pattern for the same RNA that makes spike protein. From this DNA, we will make RNA in the nucleus, and then we'll be back to normal. So let's see what happens. So again, so far, the virus is inside the cell. It has become uncoated. Now it has the DNA in it, and its job is to bring that DNA in our cell's nucleus, where it can use the DNA machinery that is inside the nucleus to make messenger RNA. That is the next step. So here is what it does. It needs to, we say docking, it needs to dock with our nucleus. So what happens is, in our cell, so if this was our cell, in our cells, we have thousands of tiny microtubules. You can think of them as small roadways. On these roadways, special proteins or special cars are running all the time, special cars. These cars, the trucks and the cars, these cars are called kinesins. Kinesin, sin means a protein. Kine is from kinetic. It's a kinetic protein. It runs, it functions, it walks. So here is this little kinesin protein. Imagine it is a car that is on this roadway. It has this virus capsid attached to it. It walks on this microtubular roadway. We call these roads as microtubules, and it brings the virus all the way to the nucleus. Isn't this interesting? So virus came in, released its capsid, which has the DNA in it. Now the virus needs to bring its DNA in the nucleus. So our cell is going to help that we have roadways in the cell. These are microtubules. On the microtubules are small cars. These cars are called kinesins. Kinesins are going to pick up the virus itself and haul it to the nucleus. Kinesin's job is to move the cargo from one place to another place inside the cell. Okay, so once the virus has brought to the nuclear membrane, there are tiny pores in the nuclear membrane which are called nuclear pores or nuclear pore complexes. Pore simply means a hole. And we have talked about nuclear pore complexes in the case of ivermectin as well, because remember, ivermectin blocks the SARS-CoV-2's cargo from going into the nucleus through these nuclear pores. So these are similar nuclear pores. But this virus here, the modified adenovirus, it does not need importin alpha and beta that was used by ivermectin. It can actually be brought in to the nucleus and attached to the nuclear pore by the kinesin. Once it is attached here, so if you see here, the virus is now attached to this nuclear pore, and what virus does is that its outer coating, the capsid, is almost kind of disintegrating, and the nucleus that is present in it is injected through the nuclear pore into the nucleus of our cell. So now the spike protein DNA has been injected in the nucleus of our cell, where our remaining DNA is. Now what happens? What's next? From this DNA that has arrived in the nucleus, we now need to produce messenger RNA. That messenger RNA will then produce the spike proteins, as we have been seeing with the other vaccines in our previous days. So let's see how does that happen. So now imagine this huge structure is the virus DNA present in our DNA. I have not drawn our own DNA in here. So I shouldn't say virus DNA present in our DNA. That's wrong. It is virus DNA present in our nucleus, where our remaining DNA is present as well and doing its own function. Now that this DNA is in our nucleus, DNA has machinery or enzymes that can recognize a DNA and that can express a DNA, meaning they can open up that DNA as a recipe book, and then they can work on the instructions in there to help the cell produce some proteins. So imagine this whole part here, this whole thing is the viral DNA. It is present in our cell. This part here, as you can see, I have made it opened bigger. So this is called the gene expression. A part of the DNA has become opened by the machinery inside the DNA, inside the nucleus. And this was the original recipe. And then our machineries, our enzymes, make a copy of that recipe, the one in green. This copy is called messenger RNA. And we talked about in detail the structure of the messenger RNA yesterday. So imagine this messenger RNA is now formed in our nucleus by the enzymes that work on the DNA. This messenger RNA now needs to get out of the nucleus to go in the cytoplasm of the cell and work with our ribosomes to make proteins. Which proteins? Spike proteins. So what would happen is, if you see here from yesterday's talk, this is the actual recipe. On the recipe, we have a cap. On the other side, we have a poly A tail. Then we attach this whole thing to a machine, a small tractor, a small enzyme that is going to haul it outside of the nucleus. It is going to drag it out of the nucleus and bring it in the cytoplasm. So this is the cap binding complex. cap binding complex. It is a small tractor machine or enzyme that is going to drag from the cap and drag the RNA out of the nucleus. So once the RNA has come out of the nucleus, then we saw yesterday that RNA will be then attached with the ribosomes that are present in our cytoplasm. These are those chefs who are going to look at the recipe and they're going to make proteins. So look what happened. How did this vaccine differ from mRNA vaccines? mRNA vaccines directly manufactured this thing and directly injected them, and it went into cytoplasm. So nucleus was bypassed. In AstraZeneca's case, it's a DNA type vaccine. So the adenovirus is acting as a vehicle to bring that DNA into our cell's nucleus, where that DNA is transcribed. That is a medical word we use. Transcription means making a messenger RNA from the DNA. So in this vaccine, instead of making messenger RNA in the labs, we have made DNA and we have given the duty of making messenger RNA to our cell's nucleus. So the DNA is brought in here. Over there, it is converted into messenger RNA. The remaining product is the same as the Moderna or Pfizer. So that messenger RNA comes out in the cytoplasm. In the cytoplasm, the ribosomes attach with it. They make proteins. These proteins are the spike proteins. Those spike proteins, as we talked 3 days ago, they will be fed into endosomes. Endosomes are small threshing machines, small threshers. They are going to chop and break the spike protein into tiny pieces. Those pieces will be loaded up on major histocompatibility complex 1 or 2 or MHC 1 or 2, which will then present that piece of the spike protein on the cell's surface. Beautiful mechanism. So if you see here, virus brought in the DNA. That DNA eventually ended up in our nucleus. That DNA was converted into messenger RNA. That messenger RNA came out into the cytoplasm of our cell, the same cell. Cytoplasm converted that into a spike protein. Spike protein was fed into a thresher called endosome. Endosome breaks down the spike protein into smaller pieces. Those smaller pieces are loaded. Those are loaded on MIC complexes and are presented on the cell surface. Once they are presented on the cell surface, then the effector action occurs. And we have seen this many times, that it is possible that the natural killer cell or cytotoxic T cell that is attached here would become active, or it is possible that a T helper cell would become active, which would then cause B cell and cytotoxic T cells to become active. We have talked about it many times. So I just simply said here, the effect would occur. And the effect could be cell killing by cytotoxic T cell, and a broken cell would activate the immune system, or effect could be directly activating the immune system if the cell here is the T helper cell. So if it is a CD4 T helper cell, then B cell and CD8 T cells would become active. If it is a CD8 cell, then that would directly kill the cell that is presenting the vaccine. And I'm being brief here with this part because we have done like 20 talks about that. So this is how this vaccine works. Eventually there is spike protein production. Eventually there is presentation of the pieces of the spike protein on the cell surfaces. And eventually there is the production of the immune system response. That is how this vaccine works. There is a slight difference from Moderna and from Pfizer.